And we'll move on to our final speaker of this session, who is William Barrington from North Carolina State University. He's going to talk about responses to dietary patterns.
Thank you. Thank you all for being here. Some of my colleagues were concerned there might be a light turnout right now since this is right before lunch, and apparently there's some Pokemon out in the hallway. So I appreciate all of you resisting your hunger and the Pokemon. It's well known that obesity has been on the rise in the United States for several decades. And the U.S. government has promoted health advice to try to mitigate health problems from obesity and metabolic syndrome. And while the figures that they use to promote these different diets have changed throughout the years, one thing has remained consistent, and that is the idea that a certain dietary pattern will be beneficial for all individuals in a population. What we're finding is that diet response is highly individualized. And this idea of diet response being individualized is not new. Back in the 1950s, Roger Williams, a professor, suggested that individuals might respond very differently to diet. And he used this analogy of two groups. Group one, all of the individuals are phenotypically identical. And so when you look at the average response, it is predictive of the response in each individual in the population. Contrast that with group two, which the average response is the same for all, or the average response is the same for the group, but all individuals have a unique response. And in this case, the group average is not predictive of the response in the individuals. And so we want to be able to look at individual effects to diet, and we do this by modeling 4 individuals in a population using 4 inbred mouse strains chosen based on their susceptibility to various diseases. And the genetic diversity between these strains is similar to the genetic diversity between 2 unrelated human individuals. The diets we chose for this study have been previously correlated with health outcomes like the American diet or the Western diet. It's correlated with poor health outcomes, whereas the Mediterranean and Japanese diets are correlated with long lifespan and beneficial health effects. We also looked at the effects of a ketogenic diet. This is essentially a hardcore Atkins-style diet. It's high in fat and has no carbohydrate. Traditionally, it's been used for epileptics and some diabetics. Now it's been used more for weight loss as well as athletic performance. And we compare the effects of all these diets to a standard mouse chow. So we feed the diets to 10 animals per strain and sex, and we look at a variety of clinical parameters related to metabolic syndrome over the course of 6 months of feeding these diets. Here I have the first results page, and all these graphs will be set up the same way. So the baseline is the effect of a mouse on a standard diet, and these bars show change from the standard on the various experimental diets. And first, I show just all of the mice grouped together. This is how a human diet study would be performed. You take all the individuals with various genetic backgrounds, group them together, and look at the average response. What we find when we do that is that all the high-fat diets cause an increase in adiposity, the Western, the Mediterranean, and the ketogenic. This is consistent for the NOD strain. However, only the Mediterranean and Western diets cause a significant increase in adiposity in the B6 strain. We see just the opposite effect in the FEB. They actually have an increase on only the ketogenic diet. And the A strain is resistant to adiposity changes. They have just very minor changes in adiposity level. We also look at glucose metabolism. What we performed here was a glucose tolerance test, and these values are changes in area under the curve on the glucose tolerance test. What we find is, again, the high-fat diets all have negative effects when all individuals are grouped together. And this is consistent for both the B6 and the FEB strains. However, the A strain and the NOD strain do not have significant effects on glucose metabolism from any of the diets. Here we have fasting insulin levels. We see an increase For the consensus in just the Mediterranean diet, and that matches what we see in FEBs, but the most profound effect is in B6 mice eating a Western diet. And again, the A strain has minimal effects. Cholesterol levels are another important factor in metabolic syndrome. First, I'll show you the effects on total cholesterol. The consensus tells us that both Western and ketogenic diets increase total cholesterol. And this carries for most strains, with the exception of the A strain, which does not have increases on the Western or ketogenic diet. But it's very important here to note the differences in the type of cholesterol being increased. So here's a chart showing LDL cholesterol, and you'll notice that the Western diet for each strain causes an increase in LDL, but the ketogenic diet does not increase LDL. And just the opposite, when we look at HDL, the ketogenic actually increases HDL cholesterol, where the Western diet generally does not, or at least to a less degree. And so what we see here is that the Western diet generally has negative effects on cholesterol, whereas ketogenic generally has more positive effects. Here I have the adiposity graph that I've already shown you. And we looked at how metabolic rate impacted adiposity levels. And we were interested to see that the ketogenic diet increased metabolic rate in all the strains. And it was very profound in the AJ strain. And this increase in metabolic rate was independent in changes in activity. So the mice weren't just moving around more. This is an increase in basal metabolic rate. They're burning more calories just sitting in the cage. And so to go further into the metabolic rate differences that we saw in the A strain, we wanted to look at the effect of mitochondria. And mitochondria, as for a basic biology review, is the powerhouse of the cell, which I'm sure you've all heard. And what it does is it generates ATP, and it does so by creating a proton gradient. This proton gradient is used to drive ATP synthase, which of course creates ATP. However, protons can dissipate back across the inner mitochondrial membrane through proteins, including uncoupling protein. And in this case, ATP is not generated. Energy is simply lost as heat. We do see an increase in heat expenditure in the AJ mice, which is consistent with what we'd expect if there was increased proton leak. And so to actually look at the mitochondrial function, we used a tool called the Seahorse Flux Analyzer. And we examined a measure called apparent respiratory control ratio. Short story is that this ratio indicates the amount of proton leakiness, and we do see about a 15% increase in proton leak across the inner mitochondrial membrane in the liver. And so this indicates that at least some of the heat is likely being lost through this. We're looking further into what mechanisms are causing this, whether it is an uncoupling protein effect. We're also looking at other tissues, including brown fat, to see if this is happening in other parts of the body. There's been a debate in the nutrition field regarding what is the most important aspect when looking at obesity. Is it simply the amount of calories that you're eating, or can the type of diet you're eating also affect your obesity rate? And so here we have the food consumption, the amount of calories the mice were eating. And in some cases, mice simply ate more calories, and this increase in calorie consumption led to a proportional increase in obesity. However, there are other cases where mice actually had a reduction in the amount of food that they were eating, yet they still had an increase in obesity. So this indicates to us that there are specific diets in specific genetic backgrounds where individuals can gain more weight despite reducing calorie intake. And so we have a variety of parameters that we've looked at in this study. And I don't have time to go through them all today, but I wanted to give you an overview of what we're seeing. And so this chart shows the effects, the aggregate view of the clinical parameters we looked at in either all the mice grouped together or each of the strains individually. And so a blue bar here indicates that there's no change from what we see on a standard mouse chow. Yellow indicates that there's some mild negative effects, and then red means that the mice really did poorly on that specific diet. And what we see here is that the view of all individuals grouped together does not match each individual within the population. And this argues that the idea of providing nutrition, a certain dietary pattern for all individuals is not going to be as effective as providing specific nutrition advice based on the genetic background of the individual. So here's a more simplified view of which diets you would want to avoid based on the data we've collected. So on average, individuals perform poorly on the Western diet, and so this is what you would see in a typical human study, and it also matches what human studies are showing us, that on average, Western diets have poor effects. However, when you look strain by strain, you'll see some differences here. The A strain is really quite resistant to negative effects of diet, and specifically on the Japanese diet, they had this odd effect where they had detrimental effects on liver function. The B6 mice did poorly when they were fed a diet that had both high fat and high carbohydrate. But if the mice were fed just high fat without the presence of carbohydrate, their health remained normal. They had no negative health consequences. And we saw just the opposite in the FEB mice, where they did very poorly on the ketogenic diet, but did better on the Western. And the non-mice reacted poorly to any of the diets that were high in fat. So to share with you some of the findings from the study, there's no universally healthy diet in this population we looked at. The phenotypic responses are dependent on diet and strain. As we show in the photo here, on the left, there's a FEB mouse, which is not prone to gaining adiposity on the Western diet, whereas the B6 mouse is. Some of the mice were generally resistant to negative effects of diet, including the A strain, whereas other mice were highly susceptible. For example, the NOD mice that did poorly on any of the high-fat diets. We found this increased metabolic rate on the ketogenic diet, particularly in the A strain, and we're following that up. It seems that mitochondrial leak in the liver is at least partially causing that effect. And it's important to note that the mechanism of weight gain differs. So some individuals gained weight simply because they were eating more calories, whereas other individuals gained weight despite limiting their food intake. And so with that, I'd like to acknowledge all my lab members who have helped me out a great deal with the project, as well as collaborators. Those bolded have analyzed some of the data you've seen today. And we also have a number of undergraduates that have helped with the project, some of which are in the lab right now. One of the teams is doing F2 follow-ups, searching for obesity genes. That's Team Chubby Chasers. And then we also have a group that's looking at effects on microbiome. We call them Team Number 2. And with that, I would like to take any questions, and I appreciate your time.
Thank you. Questions?
That was a nice talk.
Thank you.
Did you look at the microbiome, or did you collect fecal samples, and are you planning on looking at that in the future?
We did, yeah. So Team Number 2 is actively working on that as we speak. We did collect samples, and we are currently investigating what the effect of the microbiome is. We know that they're, just from a preliminary look, we do know that there are interesting differences, both by diet and strain. And actually, we tested some mice at both North Carolina and some in Texas, and we see very interesting environmental impacts as well.
There's one back here.
Should I go?
Go ahead.
Yeah. Do you notice the differences between the sexes?
There are differences between the sexes. Some of the phenotypes were more extreme, like weight gain in male mice. Thinking of B6 male mice had more exaggerated weight gain. Generally, the trends followed the same direction, but the severity of the effect was different between sexes.
What's the specific nutritional difference between the Mediterranean and the Japanese diets?
So we went to great lengths to make these diets as closely match the real human diets as possible. So not only do we match the macronutrient profiles, that is, match them to the human diet upon which they're based. So they have different macronutrient profiles between the Japanese and Mediterranean. They also have different ingredients. For example, in Mediterranean, the main starch is wheat, whereas in the Japanese, the main starch is rice. We also include bioactive compounds. So in the Mediterranean diet, there's red wine extract included. There's green tea extract in the Japanese diet. My advisor, David Threadgill, said that we needed to find some Budweiser extract for the Western diet, but I wasn't able to do that. So yeah, we match them based on fiber content, macronutrients, ingredients, and bioactive compounds.
Here, have you considered about the activity among different strains? I think some strain may be more active in movement, but some less. Then this will affect if they will get obesity.
Yeah, and we did see that. So we monitored the activity in all of these mice, and there certainly are strain-dependent effects. The FEB and NOD mice are much more active than the B6, and then the A strain mice are known as couch potatoes. So we certainly see differences in terms of the activity level, but still, despite that, the activity level on different diets within the strain did not differ, and we still saw very different effects.
Okay, I think we'll draw this session to a close, and thank you very much. Thank you.